My reading resembled the biochemistry chapter.
Yeah.
Yeah.
Can we just take our time on this bit?
I'm guessing you recognized DNA as one of two types of nucleic acids, which remember we said was one of the four major classes of biomolecules. We said that biomolecules are just gigantic molecules, right? They're macromolecules. But they're made out of a monomer, a small piece. And it's a lot of small pieces that add together to make a big thing. What is the monomer of a nucleic acid? And we know that the polymer is the nucleic acid, which is either DNA or RNA. Do you remember the general structure of a nucleotide? It has three parts.
Isn't like phosphorus a nucleotide, and then it's a base, right?
So we have a phosphorus, a sugar, and a nitrogen base. That picture right there is the cartoon version, right? We all agree? You do have to become familiar with the non-cartoon version, but we're going to take a look at the non-cartoon version together, and then I'm going to draw over it and trace over it so you can see how we turn it into the cartoon. Why? This is one of very few biomolecules that we are going to revisit with significant depth. Why are we coming back to this one?
Because it's important.
That's the makeup of DNA.
That's cool.
It's the makeup of DNA.
So it is DNA, which is in what?
Everything. Our cells that split.
Every single cell, every single living thing has nucleic acid as its base, right? As its information molecule.
What are the three components?
Phosphate, that's where I labeled a P. So we have phosphate. And then a sugar. And a nitrogen base. Nitrogen or nitrogenous, same thing. So a nitrogen base. This is probably I don't know how to best describe the importance of DNA to you. Everything that a biologist understands to be true is based on our understanding and knowledge of DNA. It is at the absolute core of everything we teach you in biology. It's why we introduce it at the beginning of the year. It's why you heard the words or the letters DNA in grade school, right? You've all heard those before? That you're not coming here and being like, oh man, this is a crazy thing. What is this voodoo you speak? We've all heard it. It's common knowledge. But what is not common knowledge is the proper understanding of why DNA is so important. I call it the central dogma of biology. Where dogma, dogmatic, right? Belief, core foundational principle. And I will tell you the central dogma is a bit oversimplified, but that's okay. For our understanding, it's easier to peel it back to as simple as we can get and then build upon that. But basically, everything in biology is founded upon the principle that all living things contain a nucleic acid. So they contain DNA. And in a biological process, that DNA is used to make RNA. And that RNA gets used to make protein. Every single living thing uses this principle as its basis for life. Now, the reason I say this is a bit oversimplified is because if I make you think about it this way, y'all be like, that doesn't make any sense. You, as a living thing, are nothing but a series of chemical reactions that are the product of which is protein, and then the rest of you is just how those proteins interact with each other. Now, just so you know, this is some bad philosophy. Taking you and reducing you to nothing more than all of your parts, you guys recognize that you're more than just a lump of proteins, right? Yeah, right? There's something else about you. Like I can add all of your proteins together and that still doesn't make a you. That's a philosophy idea. The idea that I can break you down into nothing but proteins is called reductionism. I can reduce you to your least amount or your smallest thing. Unfortunately, biologists have a tendency to do that. So I want to warn you here. That's why I said that this is an oversimplification. Because if we oversimplify, we go down a very dangerous philosophical road. You are more than the sum of your parts. You don't look happy about that. Anybody?
I'm really confused.
Let's describe it this way. Turns out that this itself is one chapter, where we are right now. The process of going from here to there is another chapter. And the process from going from there to there is another chapter. And the process of how this makes you is a whole other chapter. So
Is this the rest?
that's from now until June.
We got June 12th.
Mm-hmm.
That's what 11
Months.
Wait, do we have?
Yes, this is a month. You are looking at a month, friend.
We should just
That's a lot.
So, I want that you have a full and complete understanding of DNA. And where's the one I want? There it is.
Can you copy paste things out of the book?
I just took a screenshot out of the book. So this is truly tiny, but I wanted to give you a picture of what the whole picture looks like, and I will focus on some things. We're going to start at the top of the picture there at DNA. And I want you to see how it adds up to make chromosomes, because that's going to be important for our understanding of how this works. Because there's a word that is often misunderstood and misused here. That one. What is a gene? This is where you're going to be like, it's what makes you who you are, right? Your genes make you who you are.
The DNA that you receive from your family.
So you know that it's passed on through family because you get your DNA from your family, right? So a gene is in fact a section of DNA. It's a section of DNA that codes for protein.
That's like the protein brain.
Consider code more like a recipe. So every gene is a recipe to make one protein. Yeah, we'll go with that. Every gene is the recipe to make one protein. And I want you to see how that adds up to make a chromosome. So remember I drew it for you in like a crazy rough sketch last chapter. But if we're looking at the top here, we have our DNA and our double helix, right? And that DNA in the double helix wraps around histones. So DNA is complex with histones. 8 histones make a nucleosome. And remember I said our nucleosomes are going to wind up looking something like a flower. And all those flowers stack on top of each other. And then they coil. And then they coil. And then that wraps into a chromosome. A gene, I need one of these that I have not doodled all over. No, that's not where no, okay, good. This is just zoomed in on that same picture, right? A gene would be a section of that DNA. So we have one gene right there, where every one of these red or green or yellow or blue things is one nucleotide. So we have one nucleotide here, one nucleotide there, there, and there. 6 billion of those total makes up all of the DNA in your body. 6 billion.
So we only have 6 billion proteins?
No. You have fewer than that because it takes like thousands of nucleotides to make one protein. So this is one gene. And then you've got some stuff in the middle that we don't know what it does yet. And then we have another gene downstream of it. So that's how you add up, or how this whole thing fits together to make protein. It's just a series of protein instructions, like a recipe book, consider it like that. And unlike a recipe book, it's not necessarily in any particular order. It's not organized by like, here's a section of genes for your hair, and here's a section for your toenails. They're all over the place. They're scattered all over throughout those 6 billion. So they're not in order. When did we start learning any of this stuff? Like when did, not like we as in us, but we as in humans. When did we figure this out?
Human Genome Project.
So we did the Human Genome Project, completed in 2001. That mapped the entire human genome, right? All 6 billion base pairs. And we are still, to this day, identifying exactly what those base pairs do. So those of you who are taking data and design this summer, that's going to be a great big part of what we talk about. How we figure out what each one of these does. Actually, we're not figuring it out. You are. Ha, have fun with that. It's going to be fun.
Wait, what was the second section? Just rectangle.
Another gene. Mm-hmm. So we had the Human Genome Project in 2001. We know that. We know it's 2000 what? Right now?
15. Human genome what?
Project. (2015), we're still figuring it out. Like figuring out what gene does what, where genes are, how they interact. It's all craziness. Where did the idea of DNA, what year can I put on that? Is this a new thing, an old thing?
Is it like 1950?
I just want to make sure we give credit where credit is due. 1952, there were some researchers trying to determine the structure of DNA, and their names are famous and synonymous with the double helix. James Watson and Francis Crick were researchers working on trying to determine the shape of DNA. And the reason that they wanted to know the shape is because that was going to tell scientists how DNA replicated. That's a huge deal, how it can make a copy of itself and be accurate. Unfortunately, there was some shady dealings in there, and there's another name I have to give you up here. They had a researcher working in their lab. It was a woman. In the 1950s, kind of a big deal to have a woman working in a science lab. She had taken a picture. It's actually it's not a picture so much as an X-ray, if you will, of the structure of DNA. And if we were to look at that picture, most of you would be like, I haven't any idea how they figured it out from that picture. Like, I'll show you the picture. What essentially happened was Watson and Crick took credit for having taken that photo, even though it was Rosalind Franklin who took that photo. The photo was absolutely groundbreaking in the name of science. Watson and Crick have gotten like Nobel Prizes for their work, and Rosalind Franklin got ignored.
How do we know she took the
When
picture?
did they find out that the girl was involved?
I want to say that it was probably the 60s before anybody really started talking about the fact that Watson and Crick took the photograph. And in their autobiographies, I do believe it was Watson who refused to acknowledge Rosalind Franklin's contribution. And I think Crick is the one who was like, you know what? We did this thing, and it was wrong. So, repentance for half of them. So biologists are always very careful to make sure that when we say Watson and Crick, we also say Rosalind Franklin. That's when we learned that DNA looked like this. And it's been since then that all of our work in DNA has taken place. So it's from 1952 until 2015 is all of everything you're going to learn from now until June. Think about all the really old people we've talked about so far. Like, we threw down some Aristotle. We talked about Charles Darwin and Lamarck. Like, we've talked about a lot of old people, Linnaeus. So these people are what we would consider modern biologists. They are, I do believe, at this point, all dead. Franklin died young. Crick died recently, I think. Well, recently, like, since I was in college. So within, like, the last 10 years. So, there's those people. They're important. Double helix, X-ray crystallography. I promised you that we had to look at look familiar? You guess so.
Yeah.
I hope it looks familiar.
Got it in 2004.
2000, there you go. See? Since college. Wow, I wish I had done something useful with that section of my brain. That one that told me when, who cares? Like, I mean, I'm sure his family cares. God rest his soul, but I could have used that brain cell. The brain cell that I used to know that he died since I graduated college. Like, I could have done something with that brain cell. You know what I mean? Like that storage space. You have a finite amount of storage space up there, and I used it for like trivia. This alarms no one else.
Well, now you have a bunch of random information that you can use at any time.
My
Can
dad
you just
has
hire an
that.
assistant to clean out all the junk you don't need, and then you'd have more space?
Isn't that in the SpongeBob? Pretty sure.
It's rough. So up in this top right corner is our nucleotide, right? Woohoo. There we go. Cartoon version, agreed? That cartoon version just shows us the phosphate, the phosphate, the sugar, this is a sugar, and our base. In DNA, the sugar that we're referring to is deoxyribose. But I don't have to tell you guys that. You knew it, and you already knew that was sugar because it ends in ose. In DNA, the sugar is deoxyribose. If we were looking at RNA, the sugar would be ribose. We call them 5-carbon sugars because the sugar is made of 5 carbons. They're pentose sugars. We can label those sugars, and we will do so momentarily. But the important part here, besides our 5-carbon sugar, is this nitrogen base. There are 5 different things that can attach there. In DNA, we use 4. Adenine, thymine, guanine, and cytosine. So we abbreviate them A, T, G, and C. There are 4 different options. And these are what they look like. So we have cytosine in the top left and thymine, top right. And you'll notice that in their structure, they are 1 ring big, right? 1, 1. So when I refer to a ring, and in chemistry when we refer to a ring, that's what we're talking about. It's either going to be a pentagon or a hexagon or an octagon. It's just a carbon backbone folded back upon itself. So there's 1 ring. Adenine and guanine are made of 2 rings. Here's 1, and here's the second. In guanine, 1 and 2. All these pictures is the chemical structure of these things. If we were to examine exactly what chemicals they're made out of and exactly how they're bonded to each other, that's what this picture is. We class them into 2 different groups. They are labeled here as such, the purines and the pyrimidines. All that is words that we use to refer to the size of the nitrogen base. So cytosine and thymine are pyrimidines. Pyrimidine is the vocabulary word that means it's a nitrogen base that is 1 ring large. So it's the nitrogen base that's 1 ring large. Purine refers to nitrogen bases that are 2 rings wide. So 1, 2, 1, 2. Those are our purines. It's kind of silly, but here's how I always remembered it. Pyrimidine is a very long word, and it goes with the smaller structure. So pyrimidine, the really long word, goes with the small structure. And purine, the really short word, goes with the bigger structure. So cytosine, C and T are pyrimidines, A and G are purines. The only difference, if I were to talk about RNA, is going to be changing the sugar to ribose, and instead of thymine, we have uracil. That's the only change. We have a U instead of T. So that's where our 5th base comes in. But for right now, we're just talking about DNA. You already understand that nucleotides have to attach to each other to make the polymer, right? Take, what do I have? Double helix.
Oh, we got P, S, and B.
I love
Picture
that
lady.
name.
And picture lady.
Picture lady.
Nucleotide
Steeler guys.
Steeler guys.
You got it. It's an E.
We got these.
But mine.
Steeler guys.
It's made up of genes.
It's a series of genes
Held by a hydrogen bond.
held together by hydrogen bonds,
Diaper by a phosphate bond.
phosphodiester bonds on the sides, Andrew.
There's proteins in it.
They have a phosphate backbone.
Phosphate backbone.
Wait, what's the word phos at the bottom? Phos-
Oh, phosphodiester bonds. What else do we know about it?
There's pyrimidine and purine.
So some of these things might be from reading last night, right? Some of these things you might recall from chapter 2. But the general idea, I think I also heard the words ladder. I think I heard the word steps. How many of you are like, yes, I have heard that it looks like a twisted ladder, where the steps of the ladder are the nitrogen bases? Like, I am familiar with this. Okay. No. That was just in there for comic relief for a moment. Definitely walruses. Really, they're just here because they're such improbable animals. Like,
I would love to ride one of
those
those.
are pretty special, right? Looking at them for a moment. Looking at them.
How old do they have to be to get the little,
The tusks? I don't know.
the little?
They have like mermaids. I don't have wings.
Yep, they definitely have flippers.
Can you imagine trying to do
I got you covered.
Is this giving your brain one moment to relax?
Yeah.
Those taste good.
I'm just imagining that.
Look at that love right there. Look at that mother parenting.
Pretty sure
Maybe
the
it's-
mother's about to kick them out of that restaurant.
Yeah.
Maybe there's no.
Aw. Okay,
Okay.
there we go.
You were young. If there was a shooting machine.
So, I told you guys I was confident you were comfortable with the cartoon version, right? So I want you to see what the non-cartoon version looks like. As we look at it, we have one nucleotide that I've highlighted here. A sound effect coming now. There's one nucleotide, right? One phosphate, one pentose sugar, and one nitrogen base. So there's one nucleotide. You'll notice, though, this picture comes labeled with this weird 5'. That's what we call the apostrophe, is prime. So we have to figure out what that means. At every one of these corners, right there, at every one of these corners, there is, these corners, there's a carbon. And we start over here, and we label the carbon. 1, 2, 3, 4, and 5. Anytime you see an angle in a picture of a chemical, and there's nothing labeled there, the angle is assumed to be carbon. Labeling our carbons, here's the first one, here's the second one, there's the third, fourth, and there's our 5th carbon. So when we look at a nucleotide, we label the sides of the nucleotide, but instead of saying like top or bottom, we call top and bottom 5 or 3. 5 here is the top, right? Right there. And 3 is the bottom. So if the strand of DNA ends with carbon number 5, we call it the 5' end, because that's the carbon that's at the end of that strand. Directly across from it is going to be a complementary nucleotide. Now, since this nucleotide here is thymine. Thymine always pairs with adenine. They're complementary, they fit together. Think about it like complementary angles, not like, oh wow, you look fabulous today. But instead, like complementary, they add up to make a straight line, agreed? You also recognize that they're spelled differently, correct? Like, oh, you look fabulous is with an I, and oh, you add up to make a line is with an E. Agreed? We all know what I'm talking about? There's a big difference. Consider this the straight line version, and they do in fact add up to make a straight line. They are complementary. We have T pairing with adenine directly across from it. But let's look at the carbons. Starting with where the nitrogen base attaches, there's carbon number 1, moving to carbon number 2, carbon number 3, carbon number 4, carbon number 5. In this instance, carbon number 3 is the one on the end. So you see how they're opposite, they're oriented opposite of each other? So this one is oriented in a different way than this one. And you could also tell because of where their oxygen is. One's on the top and one's on the bottom. This idea of them being oriented opposite of each other is absolutely essential in understanding DNA replication. It works kind of funny because of this. So you all see what I'm talking about when I say 5' and 3'.
Yes.
We all get it? Starting, what carbon do I start with when I label? But how do I know which one is carbon 1? It's the one that's connected to the nitrogen base. As I look at the rest of the chain, which here my chain is a whopping 2 nucleotides long, it's very complicated. You'll notice if it's 5' up here, then it's 3' at the bottom of that chain. And on this side, if it's 3' up there, it's 5' on the other side of the chain. So it goes from 3 to 5 there and there.
So is everything just like flipped?
It's like a perfect inverse, like a mirror image, right? So it's opposites. This is what gives DNA the name or structure anti-parallel. Because parallel means runs in the same direction, right? Parallel lines run in the same direction. Instead of thinking about them as parallel lines, because they do go in the same direction, they're never going to cross, are they? I mean, look at them. Think about them as a vector. What's a vector?
That was the that one movie.
Yes, it's in Despicable Me. That's his name, right? Vector. Having both. Really? Nobody? So it has direction, one going that way and one going that way. And since they have direction and they are opposites, instead of being parallel, they are anti-parallel. Instead of being parallel, they are anti-parallel. Seems like a really tiny detail right now. You're like, this is the smallest thing I could focus on. Agreed, except it affects the way that DNA replicates. And we didn't know that until Watson, Crick, and Franklin. We had no idea. Thanks, Watson, Crick, and Franklin. Oh, look at the manatees. Manatees? Oh, manatee. I will also allow you to call them a sea cow. They're also called sea cows.
I think that's a better name.
For dugong myself.
Dugong? Wasn't that a Pokemon? What?
I have no idea.
It was like,
I don't know what.
I used to know.
But focusing on the image that we're actually supposed to be focusing on. I can't make it big enough to, oh, you know what I can do? Make you small. Ah, gone now. focusing on the image we're supposed to be focusing on, we have one side of a DNA helix here. That's the other thing about a double helix is it occurs in pairs, right? Double, 2. This is half of our 2, where we have, I like this image as well because it does us the awesome favor of showing us all our carbons. So there's carbon 1, 2, 3, 4, there's carbon 5. And when we say phosphate, this is what we look at.
So it's a phosphorus and 4 oxygens?
Yep, every time you hear the word phosphate, it is a phosphorus with 4 oxygens attached to it.
So
That's what that word means. Yep,
what's the difference between like the equal sign and then the dash?
it's the number of electrons they share. So here are 2 electrons, and there are 4 electrons.
Oh, okay.
You'll notice that they do the same thing here. There are 4 shared electrons and only 2 shared electrons. So remember I said, you know, you're familiar, probably comfortable with your cartoon version? Well, does that look like what you've seen before for phosphate? We just give you the general shape, right? And then does that look like what I've drawn for you for the sugar? And then there's the bond between them. Here's the bond leading us to our nitrogen base that, of course, we can find our nitrogens in. So that's where we get nitrogen base. And those are usually just drawn as like a rectangle with like a shape on the end. See how our cartoon fits with the real version? Real version's actually way more important than the cartoon. Sorry, you've been lied to for a while, but nobody thought you were smart enough to do it. I think you're smart enough to do it, though.